Hi, this is Mrs. Pugh.
And Mrs. Pasley.
And we can't see him, but Mr. McDowell is here. He's just homesick today. And we wanted to talk to you about how to make your models.
Yes, for your project that we're going to be doing instead of our test for our DNA unit.
Yes. So one of the things that you're going to be making is a model of DNA that shows the nitrogenous bases, show that they're paired according to Chargaff's rules. And we have one way that you can do it, and then we'll talk about some alternative ways. So I'll let you talk about one with our first way here.
So the first way is on Axis, you're going to have these templates of our DNA nucleotides available. If you want to do it this way, you can print them at home and then cut them out. And me and Mrs. Pugh have cut out a few already. We have adenine,
Cytosine,
thymine,
and guanine.
So you can cut these out at home and use a background template for our backbone. We chose black construction paper to represent our deoxyribose sugars and our phosphates. And you can start building your DNA strand. Remember, it's double-stranded. According to the correct base pairing rules, start building your strand this way. Or another method would be.
Whatever method you would like. Now, one thing we want to caution you is we have created a document so that you work backwards from the messenger RNA.
Yes.
Because by the time we get to protein, we need you to have the messenger RNA start with a start codon. So we worked you backwards in the worksheet with the start codon for the messenger RNA to one strand of DNA to the other. So don't just start building, do the worksheet first.
Follow the directions in the project first.
If you don't want to use these paper ones, we were thinking you could use whatever you wanted to use to make your model. So if instead you wanted to use pipe cleaner with beads and then have some kind of code key that represents, yellow is guanine and blue is thymine or whatever, you could do that. As you start thinking through what you want to use for your ribosome or your transfer RNA, just common household things, we were thinking that a fork could represent a transfer RNA, and it has 3 anticodons, so I'm going to cut off one of the spines of the fork. Let's watch this, make sure I don't poke this. There it goes. And so now it has 3 spines on it. You could use something to represent your amino acid up here, and here's your ribosome. Be creative. You don't have to use this. We're just giving you some ideas.
Yep.
Yeah. So one of the things you might want to think about doing is gathering some supplies around just so that you have them. Things like scissors and tape and
Cotton
sticks.
balls.
Cotton balls could represent something, because and in your worksheet you're going to have to think of what you're going to use for your DNA polymerase and your RNA polymerase.
Yep.
We're excited. We can't wait to see what you guys create. All right.